Okay, so we're going to finish up biochemistry, talking about proteins and nucleic acids. I wanted to introduce you to Max, who's my little, I guess, emotional support animal in the office today. I'm actually dog sitting for a friend of mine. So, very handsome, getting older, German Shepherd, right? Good boy. Okay, so let's get back to what we were talking about. We've covered carbohydrates, we've covered lipids. Now we're going to finish up talking about proteins and then just a tiny little bit on nucleic acids. Okay, so the class of proteins, that's the polymer. Okay, so that's the big class, and they are made up of subunits called amino acids. So amino acids are the building blocks of proteins, and once again, don't be paranoid that I'm going to put this structure on an exam or a quiz and make you label it. I just wanted to show you this because every amino acid has pretty much the same structure on the sides. Okay, they always have a nitrogen with 2 hydrogens and then what we call a carbolic acid group. What's different between amino acids is what we call an R group. So for example, the simplest amino acid, which is glycine, has just a hydrogen there. And then as we go up, they get more and more complex. So in our cells, there are 20 different kinds of amino acids, and we use those 20 different amino acids to make whatever protein we need. And so you can get a really large variety of proteins. I mean, when you think about it, the English language has 26 letters, and look at all the different words we can make. Okay, so really you can make an infinite number of proteins from 20 different amino acids. Okay, so proteins are made up of amino acids that are joined together, and this is where those side groups come in handy, because when you want to take 2 amino acids and join them together in a protein to form a protein, you just remove a molecule of water. Okay, so we see H2O here, and you make a protein which has what we call a peptide bond. So this process is called dehydration synthesis, where we're removing a molecule of water, and it's the way to take amino acids, join them together so they form proteins. The opposite is if you have a protein and you want to break it apart into amino acids, all you need to do is add a molecule of water, and we call that hydrolysis. Okay, so one thing also that I want you to understand, and I think I'm going to type these in some notes that I'm going to upload. Let me make a reminder of that, is that proteins are the most numerous type of molecules in your cells, and they do the majority of the work in your cells. So, for example, if your cell needs a particular carbohydrate, you have proteins that will make that carbohydrate. If you need a particular lipid, you have proteins that will make that lipid for you. Okay, so there's lots of different kinds of proteins, and they really do the majority of work in your cells. So they're really, really important. So pretty much everything in us is basically made of protein. We have structural proteins. I mean, even when you look at cells, your cells actually have a little tiny skeleton. We call it the cytoskeleton, and that's made up of proteins. Anytime your cells contract, like for example, in muscle fibers, you have contractile proteins. A lot of molecules in your cells that are important to physiology are kind of big babies, like hormones and things like that. And so you have proteins that help transport them, whether they form a membrane channel in the cell membrane, or they need to hold their hand and, take them to wherever they need to go. Those are called chaperone proteins. Enzymes we're going to talk about separately, so I'm going to leave those off for just a second. But then you also have buffering proteins. Like if you've ever seen someone start to hyperventilate, you actually have buffering proteins that will rush in and keep your blood pH very constant. And also antibodies, which we're going to get into in the immune system towards the end of the semester. Antibodies are big proteins that are shaped kind of like lobsters, and they have lobster claws. And those are the proteins that will grab onto bacteria and viruses and help clear them out of your system. So they're very important in your immunity. Okay, the other thing that I want to emphasize is that protein shape is absolutely essential to protein structure. Okay, so proteins must be in a certain three-dimensional shape to function. And when they act on something, you can think of it as a lock and a key analogy. Okay, so a key has to have a certain shape to fit into a lock and to be able to unlock it. And if the key gets misshapen or it's kind of screwed up, it's not going to fit into the lock, right? So proteins are the exact same way. And when proteins start to lose their shape, we call it them becoming denatured. And so when denaturation starts and the protein starts to lose its shape, it's not going to function to its full capacity. And if it gets to the point where the protein becomes completely denatured and has totally lost its shape, it's not going to function at all. So for example, let's think of, let's say you're working in the emergency department and someone comes in who's been out in the really high heat and humidity, and they're not, hydrated properly. What's happening to them on a cellular level is that their proteins are starting to denature. And like I said, proteins do the majority of the work in your cells. And so if your proteins are getting all messed up and unwound, the reactions that you need to keep your cells going are going to start to kind of fall apart. And this can get to the point where it's irreversible. The nice thing is that if you can rescue the protein in time, it will actually renature, which means that it will go back to whatever its three-dimensional shape was. So the main type of stimuli that can cause your proteins to denature are high heat. Okay, notice it's heat. Cold will not denature your proteins. It will cause the cellular reactions, the rate of them to slow down, but cold will not cause your proteins to denature, only high heat. But also if you mess with your blood pH, that can cause denaturation, and also changes in salt or electrolyte concentration. So when a patient gets really dehydrated, what's happening is their proteins are becoming denatured, okay? Luckily, if you get an IV bag on them and you get them rehydrated, a lot of times those proteins will renature and come back to their proper shape. Okay, so the fried egg example, and even though it's kind of cliche, it really is the best way for me to explain protein denaturation. Okay, so say for example, you get up, you're kind of hungry for breakfast and you're like, you know what, I'm going to fry an egg. So you heat up your pan, you take an egg, you crack it, it goes into the pan. So when an egg is at its natural state in the refrigerator, the egg white is clear, right? And it's really runny. But as you heat it up, it becomes solid and white. Then if you decide, you know what, I'm not really craving that egg anymore, I'm just going to put it back in the fridge. When it gets to this point, if you take this fried egg and you put it back in the refrigerator, it doesn't go back to being runny and liquid. It's still solid and it's still white. That is irreversible protein denaturation. And that, if it gets to that point with a patient, that's when they're really in trouble, okay? And you can start seeing, them starting to have seizures and multiple organ shutdown. And it's simply because the proteins in their cells don't work anymore. And so, the reactions that need to happen aren't working. And yeah, that patient is in a lot of trouble. And I'm going to add some notes about that, okay? So One class of proteins that are going to be really important, and we're going to talk about them a lot in terms of microbes and how they reproduce, and then eventually how we can kill them, is a class of proteins called enzymes. Okay, so enzymes are what is known as biological catalyst. And a catalyst is simply something that speeds up a chemical reaction, up to 1 billion times, but the catalyst itself doesn't change at all. So you can have catalysts that are things like magnesium, nickel, that are actual metals, but enzymes are called biological catalysts because they're made up of protein, which is a biological, type of biomolecule. So as usual, like any protein, enzymes work in a lock and key fashion. And here's the thing that's really, really interesting. And for me, it doesn't really make the greatest sense when we think of, how can we design organisms to last and, a long time. In your cells, one enzyme catalyzes one specific reaction. And there's no other enzyme that can come in and kind of help out. Or, if the one enzyme is denatured, there's not another one that can replace it. So this is when we start talking about genetic disease. A lot of really bad genetic disorders are when a patient has a mutation in a particular enzyme that is early on in a developmental pathway. Because basically, if reaction number 1 is messed up, everything down the line is going to be messed up too, because one enzyme catalyzes, one specific reaction. So keep that in mind. If we can mess up one enzyme, you're potentially going to mess up a bunch of different reactions. So here's how enzymes work, okay? So the enzyme is the big blue thing, and then what you see labeled S1 and S2 are what we refer to as substrates. Substrate just means whatever the enzyme is acting on. And enzymes can break apart, one chemical into two. They can take, for example, on this slide, it shows taking 2 different things and then joining them into one. And so the enzyme with its particular shape encounters its 2 substrates, and then it kind of pushes them together so they react, produces a product, and then it releases the product. But notice that the enzyme remains unchanged. And that's really, how enzymes catalyze reactions. So these chemicals would probably react even if they didn't have an enzyme, but what the enzyme does is it makes sure that they come in close contact. Maybe it, squeezes them a little so that their little bonds become reactive. So it's not changing anything that wouldn't naturally occur, but it just makes sure that it's done a lot faster. That's what enzymes do. So once again, enzymes work in kind of a lock and key fashion. They have what is called an active site, and this would be kind of like the lock and the substrate is the key. So yeah, if you start to denature the enzyme, this active site is going to change shape. And so maybe the substrate doesn't fit in here as well, and the enzyme doesn't work like it should. So enzymes can take one thing and break it apart into 2. Other enzymes will take 2 separate substrates and put them together in one. It just kind of depends on what the enzyme is designed to do. But if it starts to denature, that's when you have a problem where the reaction doesn't proceed like it normally should. And if you get to the point where it's irreversible, then the reaction isn't going to happen at all. Okay, so that's proteins. Now we're going to wrap up this lecture talking very briefly about nucleic acids, because like I said, in Unit 2, we're going to get to this and give them their own separate chapter. So all I want you to know for, exam 1 and then for the quiz on this chapter 2 is that there are 2 main classes of nucleic acids, and they are DNA and RNA. Boop. Okay, that's all you need to know. I do want to mention one other very important molecule, which those of you who have taken A&P, I'm sure you've learned a lot about this, but is the molecule ATP. And the reason I mention it here is because it's very similar in structure to a nucleic acid. So what ATP is it's basically money for the cell. Okay, so you know this, when you go into a store, let's say I stop at Hy-Vee on the way home. If I load up my cart and then just take it out to my car, I'm going to get arrested for shoplifting because I didn't pay for it. Okay, the same thing happens in your cells. Anytime a cell wants to do something, whether it's, let's say, oh, we have to build a lipid, or we have to make sure that this hormone gets to where it needs to go, the cell needs to fork over ATP for that to be able to happen. Okay, so your cell is constantly producing lots of ATP, fancy name adenosine triphosphate, and every time it's going to do work, it has to burn some ATP to be able to do that. So ATP is simply the major source of energy in the body. Interesting fun fact, and I only mention this because I also teach forensic science, is when ATP starts to slowly be depleted after a person dies, that's actually what causes rigor mortis, or when the muscles become really stiff. And it's why rigor mortis develops over time. It doesn't happen instantaneously because the ATP after your cells die starts to deplete slowly. It doesn't happen instantaneously. Okay, so that's what ATP does. So ATP looks like this. This is very similar to a structure of actually RNA. But what ATP does is it takes 3 phosphate molecules, which all have a negative charge, and negative charges repel each other. So these molecules do not want to be close to each other at all, and forcing them to be close to each other creates what we call a high energy bond. So when you cleave this 3rd phosphate, it releases a burst of energy, and that's what allows, whatever your cell is trying to do to be done. Okay, so to summarize that, ATP is the main source of energy in your cells. It's similar in shape to a nucleic acid, but the 2 main classes of nucleic acids are DNA and RNA. Okay, so that ends chapter 2. I'm also going to upload some notes about biochem, just some things I want you to know, so look for that in Talon as well. And I'll put it under the, Unit 1 heading, so you'll be able to find it. Okay, thank you very much.